The last severe acute respiratory syndrome coronavirus 2 pandemic, which began in Wuhan, China, is the seventh human coronavirus infection or outbreak. It occurred as such a devastating proportion, with over 437 million people affected, with over 9.5 million deaths. Beyond expectation, Africa was least hit, with the lowest transmissibility and virulence. It also has the lowest morbidity and mortality. It is imperative to determine the reason for the low transmissibility and virulence in sub-Saharan Africa, which could be a target for drug discovery. 400 sequences isolated from France in 2020, as well as 245 genomic sequences from Nigeria, was downloaded and analyzed with various tools available at the Bacterial Virus Bioinformatics Resource Center. These sequences represent each of the region. The France sequences were meant to represent Europe, while Nigeria sequences from Nigeria was used to represent sub-Saharan Africa. So we choose France because of the centrality to Europe. And also, Nigeria was chosen because of the relative landmass, which encompasses most part of West Africa. Sequences were analyzed for phylogenetics using virus tree, a tool available at the Bacterial Virus Bioinformatics Resource Center, using the Wuhan strain, which was isolated in 2019 as a reference strain. Genome alignment was carried out in the BVBRC resource using MAUV. Variation and rotation and assembly analysis were also done. Corresponding open reading frames and protein analysis was carried out. Results revealed that most of the sequences from France had 100% identity with the Wuhan 2019 sequence, with little divergence along the genetic distances. Less number of clusters were seen as compared to Nigerian sequences with resultant heavy clusters. The phylogenetic tree for Nigerian sequences revealed that only one cluster was closely related to the Wuhan sequence. This revealed that most of the sequences were imported into Nigeria from different sources, or from different sources, but they have diverged away from time from the Wuhan 2019 strain or sequence as they cross into the sub-Saharan Africa. Analysis reveals several open reading frames coding for different proteins, but most of the striking ones are ORF3b, 3D, and 9C, with no evidence of expression in Nigeria, but expressed ambiguously in France sequences. There were also striking mutations in the ORF1A and AB as compared along the line of the tarsals in the spike proteins. These mutations, which may have occurred as the virus crosses the geographical barrier, may have been responsible for the low level of expression and virulence, as well as transmissibility as the virus crosses geographical barrier. For these studies further studies are required to exploit these domains for drug discovery. The fact that in Africa the virulence and the transmissibility became lowered is a pointer to study those domains responsible for lack of expression of these proteins for subsequent exploring for drug discovery. Thank you very much.